Hi everyone, I'm Kennedy and I'll be presenting on the study Environment Dominates over Host Genetics in Shaping Human Gut Microbiota. This was published in Nature in 2018. So someone else actually presented on this same study, but hopefully I can expand on what you learned from that or maybe provide a different perspective. After several weeks in this course, I'm sure we are now all familiar with the importance of the microbiome. Today I'm going to be talking about the gut microbiome. The trillions of microbes residing in our digestive system influence everything from digestion and metabolism to immune function and even mental health. But a key question remains: what actually determines the composition of our gut microbiota? This study takes a deep dive into whether our microbial makeup is primarily shaped by our genetic blueprint or by external environmental factors. The answer has major implications for medicine, particularly in how we approach disease prevention, treatment, and even personalized healthcare. If the environment plays a bigger role than genetics, then that means that our microbiome isn't set in stone. We may be able to intentionally alter it to improve health outcomes. But just how feasible is this? Can we truly manipulate our gut microbiota in a lasting way? With this in mind, let's begin with our bluff, or our bottom line up front. So, our key question is, what factors have the greatest influence on the human gut microbiome? And surprisingly, the environment, not host genetics, plays the dominant role in microbiome composition. And why it matters is because these findings will redefine health interventions, shifting the paradigm in microbiome research. If the environment plays the dominant role, then targeted therapies, diet changes, and lifestyle interventions are far more effective than previously assumed. This opens the doors to a from the inside out approach to disease prevention and treatment. Okay, so now that we have our bottom line up front and our key points outlined, let's continue on to the background. I want to begin the background by expanding on a few key terms regarding this study. So, the gut microbiome is the ecosystem of microbes that live in the digestive tract, and they vary in species. There's a few in the photo on the bottom right here. This community of microorganisms is a complex, dynamic population, which, when functioning properly, can offer protection against pathogens, regulation of gut integrity, and plays a role in the metabolism of energy. However, when disrupted or imbalanced in any way, inflammation and a decrease in function of the gut lining often occurs. These symptoms can activate an immune response, affecting the host's entire body and immune system. There's often a correlation between disease and dysbiosis. This correlation is explained via the gut organ axis, which is the network of communication between the gut and all the other organs in the body. This axis is critical in maintaining health and regulating overall physiological function. So, as you can see, the gut microbiome lays the foundation for the functioning of all other systems in our body. A healthy body does not exist without a healthy gut microbiome. Okay, so onto a gap in our learning. So, previous studies have suggested that both genetics and the environment influence the gut microbiota or have contradicting theories that one dominates over the other. If genetics strongly influence the microbiome, then treatments must be targeted based on genetic background. This means that this is kind of a one-size-fits-all treatment plan, and that may not work for everyone. There could be several side effects, and as many individuals respond differently to medications due to gene variability. If the environment dominates, then interventions focusing on diet, probiotics, and lifestyle approaches are more effective than previously known or believed to be. This study challenges the previously assumed concept that our microbiome is inherited genetically, ultimately determining that the gut microbiota is most impacted by the environment. This means that interventions can be more universally applicable and modifiable. Healthcare will be personalized and specific but also accessible, as small changes in diet or implementing prebiotics could achieve big improvements in healthcare. This figure here shows some strategies to improve ICI efficacy. ICI is our immune checkpoint inhibitors. So these basically measure how our immune system is functioning. And as we established before, the gut microbiome and our immune system are inherently tied. An unbalanced or dysbiotic microbiome can significantly impact the host's overall immune response, which is monitored using the ICI response rates. A healthy gut microbiome has better ICI response rates. Okay. And then you'll see in the figure, there's a lot of different strategies and techniques there in that circle. Okay. So hopefully everyone now has a better understanding of the interconnectivity between our microbiome and our immunity, and understands how determining the dominating factor of our gut microbiome determines the best approach to treatment options. While the gut microbiome's full complexity is still being unraveled, we know that its composition is closely tied to overall health. This means that if we can understand what primarily influences the microbiome, whether genetics or environment, we can unlock new ways to prevent and treat disease by manipulating its basic composition. This approach shifts the medical focus from simply managing symptoms to addressing disease at its root cause. Instead of a reactive, one-size-fits-all approach, this study paves the way for a personalized microbiome-based strategy where treatments are tailored to an individual's needs. Okay. This image here shows some connections between the gut microbiome and its impact on our other organs and systems. Reinforcing that gut organ axis we talked about earlier, this bottom-up approach could redefine healthcare. Okay. Now let's get into the article, beginning with our study design. So this study analyzed two independent cohorts, one serving as the primary and another as the replication cohort to validate our findings. So the primary cohort consisted of 1,046 individuals, all aged 18 to 70. So this is the Israeli cohort of Israeli citizens. They all had different ancestries, but they all shared a relatively common lifestyle. The replication cohort was an independent validation cohort, and that's used to test whether the findings from the Israeli cohort could be reproducible in a different population. The participants were 836 individuals from the Netherlands, aged 18 to 84. So some were excluded from the study for health reasons. For once after the study began, those health reasons became known during the questionnaire and the lifestyle beginning part. So prior to the study began, all individuals were tested medically and did lifestyle and nutritional questionnaires. Now, since the goal was to separate the effects of host genetics versus environmental factors, the researchers did this by using twin comparisons to control the variables of genetics versus environment. So the microbiota composition was partitioned into genetic and environmental components by using both identical twins who share 100% of their genes and fraternal twins who share about 50% of their genes. Okay. If the microbiome were strongly controlled by genetics, identical twins should have much more similar microbiomes than fraternal twins would, and that did not occur in this study. If twins and unrelated individuals living in the same household had similar microbiomes, this would suggest environmental factors play a major role. This did occur. Both cohorts also recorded all daily activities, including standardized and real-life meals in real time. They just logged it on their phones. And then they collected detailed dietary and lifestyle information to control what people were eating and know what was happening. So the study used several advanced techniques for microbiome and genetic analysis. Data collected consisted of stool samples, DNA samples, genotyping, lifestyle and diet records, and they were also under continuous glucose monitoring. So the stool samples were for microbiome sequencing, which used 16S rRNA gene amplification. Sorry, 16S rRNA gene 16S rRNA sequencing is the primary analysis tool of this study because the 16S rRNA gene is highly conserved across bacterial species. But it also has variable regions that allow for the differentiation of species, making it ideal for analyzing the microbiome. In addition to 16S rRNA sequencing, the study also employed shotgun metagenomic sequencing. This technique involves sequencing all of the DNA in a sample, allowing for a more total and comprehensive analysis. This provided information on the bacterial taxa present and also on the functional potential of the microbiota. And then the microbial diversity was analyzed using alpha diversity and beta diversity. So alpha diversity measured the diversity within a single sample, assessing the distribution of the microbial community. Beta diversity compared communities between different samples. The researchers used principal coordinate analysis and unifrac distance metrics to assess differences in composition between the two communities. Now that our microbiome has been properly analyzed, they did the genetic analysis. So genotyping was used to examine the host's genetic makeup and examine how genetic factors influence the microbiome composition. DNA samples were taken from the individuals, whether blood or saliva or whatever, and then they were processed and stored before being analyzed through a high throughput platform. This study used Illumina Omni Express array. The goal of this was to assess how specific genetic variants in the host were associated with variations in the gut microbiome composition. Here, they specifically looked for SNPs associated with the gut microbiota, which we'll see in our figures later. But if you go ahead and look at the photos I put up, you'll see that's an example of the twin comparison, that photo, and then the one on the right and the one on the bottom left, that has to do with 16S rRNA gene amplification and the Illumina machine. So genotyping and microbiome sequencing, as well as monitoring diet and lifestyle to establish a relatively controlled environment, enabled the researchers to investigate the interaction between host genetics and environmental factors in shaping the gut microbiota. Okay, on to our figures. So now that we know how the experiment was analyzed, let's get into the actual data. So figure 1 outlines that genetic ancestry is not significantly associated with microbiome composition. So each dot on panel 1A and 1B represents an individual participant's microbiome, and the colors represent their ancestry. At panel 1A, you will see a high statistical significance as every individual is separated based on their ancestry. It shows the diverse backgrounds of the sample, and it's very organized. Panel 1B is very different from panel 1A. This panel is much more scattered. It provides an overview of the microbiome diversity, and the low statistical significance of this graph demonstrates that similar genetic backgrounds do not necessarily have a similar gut microbiome. Panel 1C plots the Bray-Curtis dissimilarities across individuals with varying degrees of shared ancestry, again supporting that microbial composition is not inherited. Panel 1D reveals the potential heritability of specific microbial species. The analysis revealed a low heritability, but did support the idea that genetics play a role in determining the presence of certain microbes, but it's not a dominating factor. Okay. So figure 1 overall provided compelling evidence that genetic ancestry has a limited influence on the human gut microbiota. Okay, on to figure 2, which shows that genetic kinship is weakly associated with microbiome composition. So here they tested related and unrelated individuals. Panel 2A shows a scatter plot of genetic kinship versus microbial diversity for 715 unrelated genotyped individuals. Each point represents a pair, showing how related they are and how different their gut microbiomes are. The relationship is not significant, with a p-value of 0.59, meaning there's no strong correlation between genetic relatedness and microbiome similarity in unrelated individuals. Panel 2B is a distribution plot of heritability for microbiome taxa based on twins. The light green line represents total microbiome heritability, while the dark green line represents a relative abundance of heritable taxa. The overall microbiome heritability is very low, at about 2%, even when using a false discovery rate threshold of 5%. This supports that host genetics play a very minimal role in shaping the microbiome. Okay, on to figure 3, which reveals limited evidence for microbiome associations with specific SNPs. So SNPs are single nucleotide polymorphisms and are variations in DNA that occur when a single base pair is different from the standard sequence. SNPs are the most common type of genetic variation in humans and are regularly used in genetic mapping or disease research. SNPs are used in this research to allow researchers to see how individual genetic differences correlate with the microbiome. Panel 3A is a Manhattan plot of SNP microbiome associations, where each point represents an SNP tested for association with microbial taxa. The dashed lines indicate genome significance threshold. Do you see how few dots there are over the dotted line and how the majority are below that line? This shows that very few SNPs reach the genome-wide significance threshold, like they all stay below. The genome-wide significance threshold is a specific p-value of 5 times 10 to the negative 8, which needs to be used to determine if a genetic variant is statistically associated with a trait. In this case, the graph shows there's limited individual SNPs that can strongly influence the microbiome composition. Panel 3B is another Manhattan plot, but this one focuses on SNPs that have been previously linked to microbiome composition. Only 7 out of the 225 previously linked SNPs replicate at a p-value of less than 0.05. 2 of those 7 are linked to the LCT gene, which does influence microbial composition, and that has to do with lactose metabolism. But according to this data, the majority of SNPs previously associated with microbiome variation are actually not strongly associated with microbiome composition. Okay, so from our breakdown of the study and the figures, we see that the data supports that genetics have a very minimal effect on the microbiome and that there's limited evidence for specific SNP microbiome associations, supporting that environmental factors play a much larger role in shaping the gut microbiome. Ultimately, this study opens the door to controlling health from the inside out, not just treating disease after it emerges, but actively shaping a healthier microbiome to prevent disease before it even begins. Some implications of this study are early diagnosis, preventative medicine, minimized side effects, and targeted treatment. Early diagnosis is key to successful healthcare, and minimized side effects and a more targeted treatment plan will improve patient outcomes. This study is well designed, but relies on a reliable narrator. Some limitations include self-reported data. There is a little bit of self-reported data in this study, so the researchers are depending on the participants to follow the guidelines and answer honestly in the questionnaires prior to the study begins, as well as the logs during the study, in order to receive accurate results. I do hope to see this study expanded larger, though. A larger cohort allows for a more accurate representation of the population, and in this case would give further ancestral diversity, which can be used to validate these findings. Both cohorts in the study produce similar results, but a larger sample size would only reinforce these findings or find faults that a smaller size could not produce. So, in conclusion, host genetics have a minimal influence on microbiome composition, while environmental factors play the dominant role. Even in two geographically and ancestrally distinct populations, as seen from our two independent cohorts, the same pattern emerged. Genetics had a minimal impact and environment played the biggest role. Okay, here are some discussion questions. Um, and I'm looking forward to hearing back from all of you guys. Here are my references, and thank you very much. I hope you guys enjoyed it.